Hello, everyone. Okay, so we'll continue with our cell concept. Okay, now we'll focus more on what we call cell division. Okay, so when we are doing our cell concept, we learned about the two types of cells. We had what we call prokaryotic cells and what we call eukaryotic cells. Okay, so we said these two types of cells, they are able to divide and give us new daughter cells. And we made mention that prokaryotic cells are able to divide through what we call binary fusion. Then when it comes to eukaryotic cells, they are able to divide through what we call mitosis and meiosis. Okay. So these are the types of division which happens in your prokaryotic cells and the eukaryotic cells. Now we'll focus more on eukaryotic cells, where we are going to talk about the cell division in these eukaryotic cells, which are mitosis and meiosis. Okay, so that's what we have. Okay, so if we were to define cell division is simply the process by which our parent cell is able to divide to give us a new daughter cells. So we have a parent cell which is able to undergo cell division, okay, and give us new daughter cells. Okay, so now we can have, depending on the type of cell division we have, okay, so we can have two daughter cells being produced or more daughter cells being produced. Okay, so now this type of cell division, if you are focusing more on eukaryotic cell, is in two forms. Okay, we have what we call mitosis and what we call meiosis. Those are the two types of cell divisions we are going to discuss now. Okay, now before we dive deep into discussing these types of cell division, let's discuss some terms that we need to know. Okay, so we need to know what a gene is. Okay, so a gene is simply a basic unit of heredity. Okay, so that's what a gene is. It's a basic unit of heredity, and this gene is able to code for a specific trait. Okay, that's what a gene is. Okay, you also need to understand what a locus is. A locus is simply the location of a gene on a chromosome. That's what a locus is. Okay. Then you have what we call a genome. Okay. So genome is simply the total heredity of your genetic material of a cell or an organism. That is what we call a genome. Okay. So the total heredity of your genetic material. Okay. Of a cell and or an organism there. That is what we call a genome. Okay. You can have what we call somatic cells. Okay, somatic cells, except for reproductive cells, any other cell is what we call a somatic cell. Okay, then we have what we call gametes. Okay, so these gametes, you can have what we call a sperm and an egg, depending on the type of an organism you have. Okay, so if it's a male, you are going to have what we call a sperm. If it's a female, you end up having what we call an egg. Okay, those are gametes. Okay, then you have what we call a chromosome. Okay, so chromosome are elongated cellular structure which are composed of the genetic material, your DNA, and also proteins. Okay, so they are the vehicles which carry your genetic material DNA in the cell. Okay, that's what a chromosome is. Okay, okay. You also need to understand what diprod is and what hyprod is. Okay, so for diprod, it's a cellular condition where each chromosome type is represented by two homologous chromosomes. Okay, so for diploid cell, you are mostly going to see this. They will be represented by 2N. Okay, so meaning for one chromosome, you end up having two homologous chromosomes. Okay, so you have this one chromosome, which is going to be composed of, you have this one chromosome, okay? You end up having two homologous, okay? So let's say this is, you have 46 chromosomes, end up having two homologous ones, okay? Which are going to have 23, 23 like that, okay? So that's what diploid is, okay? So for haploid, okay? So for haploid, it's going to be indicated by letter N. Okay, so for haploids, these are cellular conditions where each chromosome type is represented only by one chromosome, which is N there. Okay, that's what you have there. Okay, you also need to understand what homologous chromosomes are. So when we talk about homologous chromosomes, so chromosomes of the same size and shape, which carry the same type of gene, is what we call a chromosome. Okay, so these are chromosomes of the same type, same size and shape, and they carry the same type of genes. Okay, a chromatid is one of the two duplicated chromosomes connected at the centromere. That's what a chromatid is. Okay. Then a centromere is going to be the region of the chromosome where microtubules attach during mitosis and meiosis. Okay, that's what a centromere is. Okay, so it's a region of your chromosome where your microtubules are going to attach during your meiosis and mitosis. Okay, you also need to understand what a karyotype is. Okay, so a karyotype is simply a particular allele of a chromosome that is that an individual possesses. Okay, so for example, if that person is female, so the allele they are going to carry is going to be X. Okay, then if you are going to have a male, so the specific allele they are going to carry is going to be XY. So these are simply karyotypes. Okay. Okay, so those are some of the terms that we need to understand. Okay, so now let's go deep into discussing what cell division is. Okay, so we have our cell division. Okay, so this cell division is going to be divided into three parts. Okay, so if you talk about cell division, you're going to divide it into three parts. Okay, the first part is going to be called interphase. Okay, so you're going to have what we call interphase. Okay. Then the second part is going to be what we call mitosis. Then the last part, the third part, is going to be what we call cytokinesis. So these are the three parts of your cell division. So these all three parts, they carry what we call a cell cycle. So it's going to be a cycle. So you have this cell which will undergo interphase, mitosis, cytokinesis, and then you go back to interphase, mitosis. So it's a cycle. It's a cycle which is going to continue happening for a long period of time. Okay. So now, if you talk about this interphase is also going to have subdivision. Okay, so we start from the G0. Okay, G0 simply mean you have your cell at rest. Okay, then from the G0 you are going to end up having what we call G1. Okay, then from G1 you end up having what we call S phase. Okay, then from the S phase you end up what we call G2. So these are the phases which are going to occur in interphase. Okay, then you have mitosis. Okay, so mitosis is also going to have other phases. Okay, so you are going to have what we call prophase. Okay, prophase. Okay, you have what we call metaphase. You have what we call anaphase. Then you have what we call, okay, so you have what we call a telophase. Okay, so these are the phases which are going to happen in mitosis. Then from mitosis, you undergo what we call cytokinesis. Okay, so these are the phases in terms of the cell cycle. These are the phases you are going to have. Okay, so if I was to draw the cycle, it's going to look like this here. Okay, so this is what you're having. Okay, so you're saying this is this cell cycle is having three parts. Okay, so you're having the bigger part, which is going to be what we call the interface there. You can see from here to there. This is the interface. Okay, so in interface, you start from the G0 where your cell is at rest. Okay, you're going to have your G1, your S phase, and your G2. All these are in what we call interface. Okay, after your interface, after your G2, the cell will undergo mitosis. Okay, so now in this mitosis, you are going to have what we call the prophase. Okay, you're going to have what we call anaphase. You're going to have what we call you're going to have okay, so you have prophase, metaphase, okay, anaphase, then you end up having what we call telophase. Okay, then you undergo cytokinesis there. Then you go back to your G0. Okay, so these are the processes which are going to happen in the entire cell cycle. Okay, so now let's see what is happening at each phase. Okay, so we start with the interface. We are saying interface is going to be divided into three parts. We have the G1, GS, and G2. The G0 is where you're having your cell at rest. Okay, now let's start with G1. What do we have in G1? G1 is also known as the gap phase. Okay, so the gap phase. So this term gap phase, it's referring to the process in which you are filling the gap between your cytokinesis and the S phase. The S phase, that's where your DNA synthesis is happening. Okay, so filling the spaces between your S phase and the cytokinesis is what we call the G1. Okay, so in short, in this phase, you have your cell which is preparing itself to undergo cell division here. Okay, so in G1, you have your cell will prepare itself to undergo cell division. That's where what is happening in G phase. Okay, so this is a primary growth phase of your cell, the beginning of your cell division. Okay, then you go in what we call the S cell. Like we mentioned, in the S cells, that's where you are going to have your DNA synthesis. Okay, so in this phase, the cell will synthesize your what? Your DNA. Okay, your DNA will undergo what we call DNA replication. Okay, so you form a new DNA strand. That's where DNA synthesis is happening from there. Okay, then from there, from the S phase, you go into the G phase. Okay, so in this G phase, that's where you're going to have your cell, which is now going to prepare itself to undergo mitosis. Okay, so in the G phase, you have your cell, which is going to prepare itself to undergo mitosis. Okay, so again, in this phase, you have the filling of the gap between your S phase, which is the DNA synthesis, and the beginning of your mitosis. Okay, so that's what we have in there. Okay. Okay, so during this phase, you have your mitochondria, other organelles replicate, and the microtubule there, they will begin to assemble at a spindle. Okay. Okay, so these three, the G1, the GS, and the G2, all them together, they form what we call interface. Okay, so that's what you are having in your cell cycle. Okay, so from your G2, you undergo what we call mitosis. Okay, so now let's do mitosis. Okay, so we're saying we've done our interface where we've done our G1. Okay, you have the S, then you went on to the G2. Okay, now in the interface we've seen you have your chromosomes which will occur as a chromatid. Okay, here you have your centromere replicate, your chromosome have what we call sister chromatid in the interface. At the end of the interface, you are going to have your chromosomes which are going to have what we call sister chromatid. Okay, so from there now you undergo what we call mitosis. Okay, so the first phase for mitosis is what we call prophase. Okay, so in prophase, that's where you are going to have the chromosomes are going to shorten and thicken. Okay, also your microtubules are also going to shorten there. Okay, that's the first thing you're going to have. And also, the nuclear and the nuclear envelope are going to disappear in prophase. Okay, that's what we are going to have in the first phase of mitosis. Okay, from there you go what the second phase, which is called metaphase. Okay, so in metaphase you are going to have your chromosomes which are going to align at the equator there. Okay, so these are going to be attached by what we call centromeres to the spindle fibers. So that's what you're going to have. So you have metaphase. From prophase, you've aligned your chromosome at the center there in metaphase. Okay. Then from metaphase, you undergo what we call anaphase. Okay. So anaphase, you have your spindle fibers, which will pull your chromatids apart. Okay, so you are going to have your sister chromatids, one at the end of the pole and the other one at the end of the pole. So they'll pull those chromatids towards the end of the pole. Okay, the poles. Okay, then in telophase, remember in prophase where the nuclear envelope and the nuclear envelope disappeared. So here they are going to deform in telophase. Okay. Then you're also going to have your chromatids which are going to uncoil and lengthen, okay? And then we are going to form what we call chromatin. Okay, so here your spindle fibers will disintegrate, okay, and your centromere will duplicate. Okay, that's what you have in telophase. Okay, after telophase, you undergo what we call cytokinesis. Okay, so now depending on the type of the cell you're having, okay, so if it's an animal cell, you're going to have the formation of cleavage fuel to divide in cell. Okay. Then in plant cell, you are going to have the spindle fibers in the equator to form what we call protoplasm, where the cell parts are attached. Okay. So that's what we are going to have in cytokinesis. Okay. So that all okay. So whatever we've discussed up to here define what we call our cell cycle. Okay. So that's what we have here. Okay. Okay. So now we have done mitosis. We've looked at the phases of mitosis. Okay. So this mitosis will occur mostly in plants. Okay. So in plants, you're going to find what we call mitosis. Okay. So if you're having gametes, let's say animal cells, okay, these are going to undergo what we call meiosis. Okay. So in meiosis, remember in mitosis, you end up having two homologous chromosomes, which are going to form at the end, which are two N, okay? Then in meiosis, you notice that at the end, you are going to have four daughter cells, which are going to be formed with the haploid chromosomes, which are indicated by your N, okay? So in mitosis, you have the diploid chromosomes which are formed. Okay, in meiosis, you are going to end up having the haploid chromosomes which are going to be formed at the end of your meiosis. Okay. Okay, so in meiosis, you are going to have the process in which daughter cells are going to be half, okay, the number of chromosomes as that of the parent cell. Okay. So this will occur in gametes, in the sperms and the eggs, okay? So you end up having haploid chromosomes, okay? So this meiosis is going to be divided into two parts, okay? So we're going to have what we call meiosis 1 and what we call meiosis 2, okay? Okay. So just like in prophase, you are also going to have what we call just in like in mitosis, you're going to have what we call those phases. You're going to have prophase, okay? You're going to have metaphase, okay? You're going to have what we call anaphase, okay? You're also going to have telophase, okay? Now the only difference is that here you are going to have telophase, prophase 1 and 2, okay? So this will occur twice. Okay, so we have what we call prophase 1, prophase 2, metaphase 1, metaphase 2, anaphase 1, and anaphase 2, and also telophase 1, and also telophase 2. Okay, so those are the type of divisions we are going to have. Okay, now let's discuss this one by one. Okay, so we start with the prophase 1. Okay, so in prophase 1, you have in your homologous chromosomes, which will become paired. Okay, then here, that's where crossover is going to occur between these homologous chromosomes. Okay, so take note of such statements. So crossover will occur in the prophase 1, okay, of your meiosis division. Okay, after you've done your crossover, you go to what we call metaphase. Okay, so here your homologous pairs become aligned in the center of the cell. Okay, metaphase, your homologous pairs will align at the center of the cell. Okay, you're also going to have the random alignment pattern, which is going to be called independent assortment. Okay, so for example, a cell with 2N is equal to 6 chromosomes there. Okay, so this could have any of the alignment pattern shown. Okay, so if you have the cell with 2N, which is equal to 6 chromosomes, you can have these alignment which can occur. Okay, so this is what we call independent assortment. Okay, then from there you go to what we call anaphase. Okay, so in anaphase, you are going to have your homologous chromosomes which are going to separate there. Okay, so that's your anaphase 1. Okay, so they will separate. So one of the homologous chromosomes, they will be on the other pole, and the other ones will be on the other pole like that. Okay, so you have separation of your chromosomes, your homologous chromosomes in anaphase 1. Okay, so you go to telophase 1. So this is not going to happen in some species, okay? But just like we discussed in telophase of mitosis, okay? Here you are going to have a situation whereby you're going to see the okay, so the occurring of your so the occurrence of your nuclear membrane. Okay, here you are going to see the occurrence of your nuclear membrane. Okay, but this is not going to happen in some species. Okay, then from there, you instead of going to cytokinesis direct, okay, what is going to happen in meiosis and like in mitosis, this is now going to go to the second phase of your meiosis. Okay, so it's going to go to meiosis 2. Okay, so here you start again. So you are going to go to prophase 1. Okay, prophase 2. Okay, so in prophase 2, here the meiosis where the chromosomes will condense. Okay, the occur the nuclear membrane breaks down and the spindle fibers form to separate to prepare for separation of the sister chromosomes. Okay, so here you end up having the condensation of your chromosomes. Okay, also your nuclear membrane will break. Okay, and your spindle fibers will form. Okay, and you have the separation of your uh, you have the preparation or the separation of your sister chromatids there. Okay, that is in prophase 2. Okay, so from here you go to metaphase 2, where now the separation is going to okay, the aligning of your chromosomes at the center. Okay, so here you have your chromosomes line up at the center with spindle fibers attaching to their centromeres. These are also you're preparing for chromatid separation. Then the separation is going to occur in the anaphase there. Okay, so the sister chromatids here will separate. Okay, so they'll pull away from each other. So one will be at the other pole, and the other one will be on the other pole. Okay, so now here you notice that after the first meiosis, we are using one cell. Okay, so we are using one cell at meiosis one. Okay, so we are using one cell. We are using this one cell. Okay, after we are done with meiosis one, you notice that we ended up having two cells here. Okay, now again, these two cells, they undergo the same phases. So now you notice that we are using two cells here. Okay, so we are using two cells. So here they separate, the two cells they separate. After the separation, you will undergo telophase. Okay, so telophase, that's where now again the nuclear envelope they appear. Okay, the spindle fibers also disappear here. Okay, then you end up having four daughter cells towards the end. So this will also form new cells. Okay, so you end up having, so from the two, you get four daughter cells which are going to be formed towards the end of your meiosis. Okay, so that's what you are having there. Okay. Okay, so that's what we have on cell division, on meiosis and mitosis. Okay, so now let's do a little bit of revision on the things we've done so far. Okay, so we have a question there. Let me just zoom it. Okay, let me just zoom it a bit. Okay, okay. So now we have a question here which is saying, uh, higher animals and plants are made up of different structures, which can be ranked in only one of the following order of complexity. Okay, so now what is the correct order? Okay, so with what we know, okay, so when we start doing micromolecules, you notice that the first thing you have the molecules. Okay, molecules. So the different biological molecules, they will come together to form cell components. Okay, cell components. Okay, so these cell components are same type of cells. They grouped. So you have same types of cells, same type of cells. You group them together to form what we call a tissue. Okay, so you have four types of tissues. You have the epithelial tissue, you have connective tissue, you are going to have muscle tissue, and also a nerve tissue. Okay, so these different types of tissues, you put them together. Remember, these tissues were formed by same types of cells. So you have same types of cells which are going to be grouped together. So if you have cuboidal cells, they are going to form a type of tissue. Okay, so same type of cells, you group them together, they give you a tissue. Now, different types of tissues, you group them together, you're going to form an organ. Okay. Now, these different types of organs, you get a group of organs, you put them together, they'll form what we call a system. Okay. Now, in a different system, you have about 12 systems in the body. You put them together, they form what we call an organism. Okay, so this is the correct way things are supposed to be. Okay, so now, meaning your answer has to be this one here. Okay, so you have molecules which are forming cells. Then cells, you have tissues. Then tissues, you have organs. Then organs, they form a system. They skip the system there. Then you have organism. Okay. So that's the correct answer there. Okay. Then you have the constricted part of the chromosome structure which holds a pair of chromatids is known as the. So this one we are just from discussing. Okay. So this is going to be okay, your centrosome there. Okay. So this is the one which holds a pair of chromatids together. Okay. Then you have this one, the last question is saying, in the mitosis process, the stage at which cytoplasm is divided into two regions to give rise to the formation of two daughter cells is known as. So this is the last part of your mitosis. Okay, so you're getting two daughter cells. Okay, So this should be cytokinesis. Okay, so those are some of the questions you can have from your test or an exam. Okay, so now if you want to get access to more of these videos, you can register with us. Let me just show you how you can register with us. Okay. Okay, so let me just do this. Okay. So you can register for more of these topics and practical questions. You can register with us. So you go uh, on our website. Okay. Let me just open the website. Okay. Okay. So you open your browser like that. So when you open your browser, you type Discrete uh, Learning Institute. If you don't, if you are using an Android phone, you can easily go on Play Store. You can download our app there. Okay, so it's Discrete Learning Institute. Uh, if you are using okay, so if you want, you can go on Google. If you don't want to use the app, you can go on Google or Chrome. You search Discrete Learning Institute. Okay, so it will bring something like that. So you click on that link, you open. Okay, then it will take you to the website. So you have a website which will bring options like these ones. Okay, so here there are options. You have where it says subscribe for a course. You can easily subscribe if you have an account. Okay, so if you don't have an account, you can create an account there. Then you are able to subscribe to a specific course of your choice. Okay, so when you subscribe, you will find the courses which are available there. Okay, so you can easily if you are subscribed, you can log in to your course of your choice. Let's say you've subscribed for biology, you can log in uh, there where it's in biology. You log in, then you should be able to find something like this. Okay, so there are lessons and tutorials. You have handwriting, summarized notes. You have tutorial sheet and assessment. You have a folder for past papers, quizzes, and exam. You have lab reports and samples. So if you want lessons and tutorials like this one, you can easily go in that folder written lessons and tutorials. So you open it, you'll find different topics which are there. Okay, so you should be able to go in the type the topic you want. You should be able to find it there on the website. Okay, so that's how you can subscribe with us. Okay, so we'll end here for today. Have a blessed evening or day.